is Lauchlan Fraser, who's presenting on two Geoscience BC supported mine reclamation research projects: post-mining restoration in South Central BC, modeling microbial and geochemical changes in topsoil stockpiles, and invertebrate response to mine reclamation in South Central British Columbia, effects of reclamation age on arthropod assemblages at the Highland Valley Copper and New Afton mines. Locke is a senior NSERC Industrial Research Chair in Ecosystem Reclamation in the Department of Natural Resource Sciences at Thompson Rivers University in Kamloops. Locke joined TRU in 2004. He's an ecologist with expertise in grassland, wetland, and rangeland ecosystems. Research in the Fraser Lab is focused on ecological restoration, biodiversity, and climate change. Locke, good morning, and over to you.
Thanks, Krista. This is going to be a slight departure from the previous talks because the work we do in my lab is mine closure reclamation. And as Krista mentioned, I'm going to be talking about two different aspects, two funded projects. And the first will look at topsoil stockpiles, and the second will look at trends in biodiversity following different restoration methodologies. So topsoil stockpiles are created in the development of mine sites. And often they are stored for a considerable amount of time. They're important, topsoil is, because they are important reservoirs of nutrients and native seed. But after a certain length of time, there might be degradation of these stockpiles, particularly at lower depths. And that's what we wanted to investigate using genomic tools. So we wanted to improve our knowledge on how topsoil stockpile height impacts soil quality and provide information to industry on how to alleviate that and improve reclamation practice. We looked at two different stockpiles, one at the New Afton mine, operated by New Gold, right outside of Kamloops, and the other is the QR mill operated by Barkerville Gold Mine up near Quesnel. These are different, quite different stockpiles. The one at New Afton is not as is a taller pile, but not as old, only about 7 years old. And the QR mill is 20 years old at the time of sampling. We were interested in geochemical changes, but we were also looking at the microbial community, and we extracted DNA, identified amplified through PCR, and used the ion torrent platform to sequence the DNA through amplicon sequencing. And looking at the geochemical data, this is a lot of information to show and hard to digest. But what we do see here is the two different stockpiles. The one on the right, the longer one, is the New Afton site, and the one on the left is the QR site. And the takeaway messages from this is that there are changes in geochemistry with depth, and typically we see a decrease in nitrate, sodium, boron, potassium, organic matter, and available phosphorus. And we do see increases in depth with copper, iron, and ammonium. Interesting interactions going on there with the nitrogen species. And it's quite explainable, where we see nitrate levels decreasing with depth, and that's the more available form of nitrogen for plant growth, and ammonium accumulating at depth. Taking a look at the microbial community, we've separated this into the fungi and the bacteria. Fungi on the left, bacteria on the right. And the QR mill is found on the right-hand side here. And the new Afton stockpile is found bunched together on the left. What we see here is that both fungi and bacteria separate very well with depth in the QR stockpile. And we find that very interesting because we suspect that this separation is partially a result of time, and over time these differences in the microbial community makeup is going to be more pronounced. And within the new Afton site, there's really very little distinction actually with depth in terms of the fungi and bacteria community, but this was a younger stockpile. Okay, so I'm now going to switch gears a bit and talk about biodiversity and looking at invertebrates. And this is mainly the focus of my master's student, but I've also included some information on how the microbial community changes. And here we wanted to monitor reclamation trajectory of invertebrates as well as the microbial community. And we used a chronosequence approach. So we focused most of our attention at Highland Valley Copper Mine, but we also have sites at New Afton. Highland Valley has been in operation for many years, and many areas and sites at Highland Valley have been reclaimed at different points of time. So this is where we can apply the chronosequence approach. We selected sites that were over 20 years old, reclaimed over 20 years ago, and then more recent sites reclaimed just a couple of years ago. And also some of these sites, we were also interested in how biosolids might influence invertebrate diversity as well as microbial diversity. And so we selected areas that had biosolids applied and areas where biosolids were not applied. And broadly speaking, looking across all of the different sites, and we had some 15 site locations that we compared, we were able to categorize according to different age categories. We did have control areas. These were reference sites adjacent to the mine site to compare against. And then the new sites were 5 years or younger, and the older sites were all the rest. As you can see, when we look at the Similarity Index, this is a multivariate assessment of how similar sites are. The older sites are more similar to the reference areas than the newer sites in terms of invertebrate community. Whereas if we looked at the biosolids and no biosolids applied, where the sites where no biosolids were applied were more similar to the reference sites than where biosolids were applied. So what about microbial diversity? Let's start with fungal diversity. Here we see that by age, the older sites are trending towards being more similar to the reference areas than the newer sites. So that's very similar to invertebrate diversity. And also for biosolids, where no biosolids were applied, the fungal diversity was more similar to the reference sites. Very similar results to invertebrate diversity, and we see similar trends with bacterial diversity as well. If we look at a multivariate analysis to look at comparatives, we see that the biosolid treatment was a significant predictor of microbial community composition, and that the trajectory of the microbial community composition moved towards the reference site. And this is for the fungal community. And if we look at the biosolid treatment, in this model, the age since reclamation is the major determiner of this trajectory. And the bacterial community, similar response to the fungal community, where the bacteria community composition appears to be moving towards the reference site with age. So in conclusion, we found that the microbial communities were associated with depth at the stockpiles, and the invertebrate communities were significantly associated with age and amendment. There were significant differences in the microbial community from the site with different reclamation strategies, and they did trend towards the reference site. And this is a good indicator that these sites are, the trajectory is in the right direction. And given enough time, we believe that the microbial community at reclaimed sites can potentially recover to the reference state. We're continuing work with our genomic data and looking at metagenomic sequences. And we're also exploring associations with the function of these microbial community, their functional role in these systems. Thank you for your attention.